Welcome back to the Deep Dive. Today we're plunging into a topic that is, well, foundational to medical progress, yet surprisingly challenging: recruiting patients for clinical trials. These trials are how we evaluate new therapies and medications, but finding the right people, the exact candidates needed for these studies, can often become a major bottleneck, slowing down life-saving advancements. We've got a fascinating, almost visionary source document for our deep dive, a research paper from 1993 titled A Methodology for Determining Patients' Eligibility for Clinical Trials. Yes, you heard that right, 1993. It's an in-depth look at how computer systems could assist clinicians in this knowledge and data-intensive task, long before terms like AI were really common. Our mission today is to unpack the ingenious solutions proposed decades ago that still resonate powerfully today, perhaps even more so with our current technology.
It's really interesting. What's particularly insightful about this paper is how it dissects a deeply human and intricate process, evaluating a patient against a long list of criteria, and then systematically builds a framework for automation. It's a brilliant example of applying structured thinking to a very real human-centric problem, recognizing that even the most expert clinician can be, well, overwhelmed by the sheer volume of information.
Right. So let's start by setting the scene.
Yeah.
What was the problem these researchers identified? Because it's still so relevant today. Clinical trials, for all their scientific rigor, depend fundamentally on patient participation. But as the paper points out, recruiting a sufficient number of participants within a defined time frame may jeopardize the successful completion of a clinical trial. This isn't just an inconvenience, it means promising therapies could be delayed or, even abandoned.
It's true. And the paper cites some stark examples that really drive this home. They noted how none of the 10 clinical trials monitored by one division of the Veterans Administration could be completed on time. And 2 of these trials had to be terminated prematurely, specifically due to insufficient recruitment. Think about that. A huge loss, not just in terms of research investment, but more importantly, for the potential medical advancements that could have emerged. Imagine the impact on patients waiting for those new treatments.
And why is it so incredibly difficult? Why is patient recruitment such a persistent issue?
Well, the eligibility criteria for these trials are incredibly detailed, specific, and often extensive. We're talking about everything from demographic details, a patient's prior or current diagnoses, specific laboratory test results like vital immune cell count, such as CD4, or blood clotting indicators like platelet count, all the way to subjective symptoms, physical findings, current or prior medications, and drug allergies. These aren't simple yes-no questions. For instance, a patient might need to be HIV positive, have a platelet count greater than 75,000 per cubic millimeter, or have not taken a specific drug like rifampin within the last 30 days. It gets complicated fast. Exactly. And what's fascinating here is that a clinician's manual process for evaluating all these points is anything but straightforward. It's a dynamic and ongoing process, often requiring multiple evaluations over time. A clinician might need to collect additional information or actively adjust a patient's treatment therapies, maybe in anticipation of a trial, or even order unusual tests specifically required by the clinical trial protocol tests that aren't part of routine care.
Right.
And you consider a large academic clinic, often running dozens of clinical trials simultaneously, each with its own complex set of rules. It's not surprising that potentially eligible patients may be overlooked. Imagine the information overload a busy doctor faces trying to keep all that straight for every single patient.
That paints a really clear picture of a truly challenging landscape. So, with that in mind, what did these forward-thinking researchers propose back in '93? The core idea was to leverage computer programs to assist clinicians. It wasn't about replacing their expertise, but like empowering them with a sophisticated tool.
Exactly. They envisioned a well-designed computer system that could, for instance, identify areas of incomplete information, clearly flagging what data was still needed. It could indicate the possibility of a patient's condition changing over time, suggesting they might become eligible in the future. That's huge. And crucially, it could highlight characteristics that can be modified or acted on to render a patient eligible, providing clinicians with actionable insights rather than just a pass-fail.
Okay.
This shifts the process from passive screening to active management. This isn't about replacing the clinician's judgment, but about reducing their immense information load and making the identification process more systematic, ensuring fewer patients slip through the cracks. Their initial findings, actually, from screening HIV-positive patients strongly suggested an automated eligibility determination program such as ours has the potential to increase a clinical trial's accrual rate.
That focus on reducing information load makes a lot of sense. But how do you even begin to automate such a nuanced process? The researchers realized the first critical step wasn't about coding, but about deeply understanding and um, categorizing the eligibility criteria themselves.
Yeah.
This is where they laid the foundation for truly intelligent assistance.
They did, and this classification is really insightful because it highlights the different natures of patient data and the flexibility involved in eligibility. First, stable requisite criteria. These are essentially immutable preconditions, things like a patient's history of a disease or maybe an intolerance to certain drugs. They're historical facts unlikely to change.
Got it. Fixed points.
Exactly. Second, variable routine criteria. This is data that's collected routinely in patient care and is relatively stable over short periods, things like a CD4 count or platelet count. These can change, but you usually get updated measurements pretty regularly. Third are controllable criteria. Now, these are conditions a physician can modify. For example, if a protocol prohibits a specific drug within 30 days, the physician might be able to substitute a similar drug. And then by waiting 30 days, the physician can change the patient's status from ineligible to eligible. It turns a barrier into a potential pathway.
Ah, that's clever. Proactive.
Fourth, subjective criteria. These rely heavily on a physician's judgment, like estimating the likelihood that a patient will comply with the protocol or their estimated survival. These are inherently, qualitative assessment.
Right, the human element.
And finally, special criteria. These require unusual laboratory tests that are not typically performed, maybe things like lumbar puncture studies. These tests might be costly or invasive and are typically performed only after a patient is identified as a strong candidate.
So what does this detailed breakdown mean for, the clinician or researcher trying to navigate these complex trial protocols? Why is this categorization so powerful?
Well, this classification serves two really vital purposes. Prospectively looking forward, the program can flag patients who only fail on variable or controllable criteria. This is incredibly powerful because it alerts the clinician that the patient might become eligible later, or that a specific actionable intervention could render them eligible. Imagine the efficiency gains proactively identifying and nurturing potential candidates.
Yeah, instead of just writing them off.
Exactly. And retrospectively looking back, this framework helps analyze why patients were ineligible. It helps identify systemic issues in recruitment or maybe reveal criteria that are just too restrictive for their specific patient population.
So, okay, once you've categorized the criteria like that, how do you actually determine eligibility when information might be missing or uncertain? Because, let's face it, patient records aren't always complete or perfectly up to date in the real world. The paper introduces two clever approaches for this. Let's start with the qualitative approach.
Right. This method assigns discrete, sort of non-numeric eligibility scores, essentially a spectrum of confidence. You've got P, pass. The patient definitely meets the criterion. Simple enough. PP, probably pass. The patient probably meets based on a reasonable assumption about some missing data. N, not available. The data needed is missing, and you can't really make a reasonable assumption. FP, probably fail. The patient probably fails, again, based on an assumption about missing or incomplete data. And F, fail. The patient definitively fails.
Okay, but missing data is such a notorious problem in real-world patient records. How did they even begin to grapple with that in this qualitative model?
Yeah, that's key. The system uses what they call missing value assumptions, which are a crucial design choice. They're built to be inclusive rather than exclusive, specifically to avoid mistakenly ruling out patients who might actually be eligible. It's about filling in the gaps intelligently.
Inclusive rather than exclusive. Interesting.
So, for instance, if a patient's medical record doesn't explicitly state they are currently in another trial, the system might safely assume that the patient is not enrolled. That's a default assumption. Or for something like a platelet count, if a previous value was well within the required range, it might assign a PP score, assuming the condition is stable satisfied, meaning it probably hasn't changed drastically. This isn't just about what's there, but intelligently inferring from what's not there or what was there previously, basically to cast a wider net initially.
That inclusive approach to missing data is key, but it always comes with a specific balance, doesn't it?
Yeah.
A trade-off. More assumptions might mean identifying more potentially eligible patients.
Right, more true positives.
But also potentially more false positives.
Yeah.
Patients who get flagged but turn out to be ineligible after all, requiring follow-up time.
Precisely. It's that classic sensitivity versus specificity trade-off. And once those individual scores, P, PPN, FPF, are assigned to each criterion, the system then combines them using logical rules, like a sophisticated flowchart, using basic A&D and/or truth tables to determine overall eligibility for a set of criteria.
Okay.
So, for example, if a patient is P for HIV status and PP for platelet count, combining them with an A&D logical rule results in an overall PP for those two. This logic provides a powerful summary statement, allowing the system to say something like, she had a PP score on the stable requisite criteria, but an F score on the variable routine criteria, clearly indicating why someone didn't make the cut that day rather than just a simple yes or no.
Okay, so the qualitative method offers a pragmatic way to handle uncertainty and missing data, casting that wider net. But for those seeking a more precise, quantified assessment, the paper explored another powerful technique, the probabilistic approach. How did that one differ in its core philosophy?
This is where things get a bit more mathematically sophisticated. It uses Bayesian belief networks, leveraging the power of probability theory. Think of them as graphical models that map out the probabilistic relationships between different factors, like different criteria, or how past data influences current data calculating likelihoods. Instead of discrete scores like P or PP, eligibility for a criterion is expressed as a probability between 0 and 1.
Okay, a percentage chance.
Exactly. Nodes in the network represent individual criteria, combinations of criteria, and even how related factors, such as a patient's previous platelet count, might probabilistically affect their current one. It's a much more nuanced, interconnected view of the data.
So you feed in the available patient data, whatever you have, and the network, with its understanding of these relationships, calculates the likelihood of satisfying various criteria, even for those without direct current observations.
Exactly. And what's really fascinating here and quite powerful is that it can compute a precise probability that a patient satisfies all eligibility criteria simultaneously. This provides a much stronger, more quantified statement than the qualitative approach can offer. It gives you a single consolidated probability number for overall eligibility based on everything it knows. It's a very robust way to manage uncertainty.
Okay, so we have these two elegant solutions, each with its own strengths.
Yeah.
But they're not interchangeable, are they? They seem to interpret eligibility quite differently, especially that summary score.
No, not entirely interchangeable, and this is a really critical distinction. They both handle missing data and summarize eligibility, but they diverge significantly in their interpretation of the final summary score. The qualitative approach's summary score, like that overall PP, is not a true probability that a patient satisfies all criteria simultaneously.
Oh.
If a patient gets an overall PP, it essentially means they probably satisfy at least one criterion that was uncertain and definitively satisfy the rest. It doesn't mean they probably satisfy all criteria with a specific percentage chance. Think about it this way. Even if a patient has a, say, 0.9 probability, a 90% chance of satisfying each of 10 independent criteria, the probability of satisfying all 10 simultaneously is actually only about 0.35 or 35%. The qualitative approach might still give a PP summary score in that situation, which could be misleading if you interpret it as a high overall likelihood.
That's a huge distinction. So the probabilistic approach offers a stronger, more mathematically accurate overall statement about true overall eligibility. Given that precision, why not just use that one? Seems better.
Well, it comes at a significant price, primarily in complexity. Modeling the complex dependencies among all the criteria in a Bayesian network, meaning how factors like a patient's overall health might influence their organ function tests, or how one lab result correlates with another, is non-trivial. It's hard work.
Right.
Clinical trials often have dozens of criteria, and these aren't always independent variables. You need to elicit distributions from domain experts, basically get detailed probability estimates from experienced doctors for all these potential relationships. That's time-consuming. And the computation itself, solving the Bayesian network, can be NP hard, meaning it quickly becomes incredibly complex and computationally intensive as more criteria and dependencies are added.
Okay, so practically difficult to scale.
Exactly. The qualitative approach, on the other hand, is much simpler, conceptually and computationally much more efficient. It's linear in the number of criteria.
So what does this all mean for practical application? How do you get the best of both worlds, maybe harness the strengths of both, without succumbing to their weaknesses?
This is where the researchers proposed a really brilliant combined approach, which I think is very insightful. You use the Bayesian belief network, the probabilistic method, to get a principled way of computing beliefs, that is, those precise probabilities, for each individual criterion. You leverage its consistent handling of uncertainty and missing data for each specific point.
Okay, so get the best probability for each part.
Right. But then you map the probabilities into qualitative scores, turning that P09 probability into a P or PP, for instance, and use the simpler logical rules, the AND and OR truth tables from the qualitative method, for summarization. This avoids the immense computational complexity of deriving that single overall probability for the entire protocol, which is the NP hard part. But you still get clear, actionable summary scores that clinicians can quickly interpret, like PP on stable, F on variable.
That makes sense.
This isn't just a clever technical workaround. It's a profound lesson in pragmatic problem solving. Identifying where precision truly matters most at the individual criterion level and where computational efficiency is paramount for the overall summary. It's a blend that's still incredibly relevant for building practical AI systems today.
And this wasn't just theory, which is what makes this paper so compelling. The researchers actually implemented the qualitative approach, or maybe the combined one, in a system they called Therapy Helper. What did they find when they applied it in a real-world scenario?
Yeah, they implemented the qualitative approach. They meticulously encoded the criteria for 17 HIV and AIDS protocols that were active at the Santa Clara Valley Medical Center, SCVMC, back in 1990. Then, they retrospectively analyzed 60 HIV-infected patients over a 7-month period, which covered over 300 distinct patient visits, comparing the system's eligibility findings to what actually happened in practice, who was considered, who was enrolled.
Okay, let's unpack those real-world results. What did Therapy Helper reveal about patient recruitment on the ground?
The findings were quite compelling and frankly a bit sobering. The system clearly showed that clinicians inadvertently missed a significant number of opportunities for enrolling patients.
Oh, wow. How significant?
Well, specifically, 12 of the 60 patients, 20%, were provisionally eligible for at least one protocol during the sample period, but were not enrolled. Think about that. 1 in 5 patients who could have potentially benefited from a trial, or at least advanced critical research, were simply overlooked.
That's substantial.
It is. And even more remarkably, 54 of the 60 patients, 90%, could not be ruled out as ineligible at some point in their care, meaning the system saw potential eligibility for almost everyone at some time point. And a massive contributing factor to these missed opportunities was, unsurprisingly, data scarcity. Data required for eligibility evaluation were frequently missing. For example, platelet counts were missing for nearly a third of all patient visits.
30%.
And CD4 counts, which are absolutely critical for HIV/AIDS protocols, were missing for a mean of 128 visits, which is 43% of the visits.
20% missed eligibility. That's a huge number of potential participants for trials. And the missing data statistic is equally eye-opening. Almost half the CD4 counts weren't available when needed for evaluation. It really underscores the immense challenge clinicians face, and honestly still face, trying to keep all this straight manually.
It absolutely does. If we connect this back to the bigger picture, it highlights the immense potential value of these kinds of automated systems. Even with relatively simple missing value assumptions, Therapy Helper could identify patients who were eligible based on the available data but weren't considered by the clinical team. This shows the potential to significantly increase patient accrual, not just by alerting clinic staff to who might be eligible, but crucially by highlighting what data is needed to confirm that eligibility, or maybe what actions can be taken, like changing a medication to make them eligible. It transforms what can be a passive screening process into an active, guided one.
This research, though focused specifically on clinical trials back in the 90s, feels like it has much wider implications for medicine, and maybe even beyond healthcare altogether.
Absolutely. The paper itself explicitly points this out. They say that clinical trial protocols are similar to medical practice guidelines, those standardized specifications prepared, developed by expert bodies to define appropriate tests and procedures for certain conditions. Both require evaluating patients against detailed criteria, and both face the same fundamental challenges of complex data, potentially missing information, and making consistent decisions.
So, the same challenges would apply. Missing data in patient records affecting guideline adherence checks, eligibility criteria based on parameters that change over time, or even clinician-controllable factors influencing whether a guideline is applicable.
Exactly. The methodology they develop for finding eligible patients for clinical trials can also be applied to the problem of determining whether practice guidelines are applicable in a given clinical situation. It's really a testament to the power of structured problem solving. Identify a core challenge, abstract it, understand its components like they did with the criteria classification, and then create adaptable solutions that can serve multiple, seemingly distinct but fundamentally similar problems.
This deep dive into a paper from 1993 really shows us that the challenges of information overload and complex decision-making in healthcare aren't new at all. But neither is the ingenuity in using technology to try and cut through it. The therapy helper system, with its qualitative and probabilistic ideas, offered a concrete, implementable way to make clinical trials potentially more efficient and inclusive decades ago. It's a powerful reminder that some of the new problems we grapple with today, especially around data and AI in medicine, were being systematically thought about and tackled by brilliant minds a long time ago.
And it really raises an important question for us today, doesn't it? In our increasingly data-rich, maybe even data-overwhelmed, but definitely time-poor world, where else are we missing opportunities? Not just in medicine, but in other complex fields, simply because of the sheer volume and complexity of information we have to deal with.
What stands out to you listening to this deep dive? Think about how many other knowledge-intensive fields could benefit from a similar systematic approach, breaking down the problem, understanding the data, classifying the decision points, and building tools that truly augment human expertise rather than just trying to replace it. Until next time, keep digging, keep learning, and stay curious.